Hi everyone, myself Dr. GSN Koteshwar Rao, Associate Professor at KL College of Pharmacy, KL University, Guntur, Andhra Pradesh, India. So on behalf of management, principal, staff and students, I take this opportunity to welcome you all for today's international webinar on Advanced Pharmaceutical Analytical Techniques by the renowned speaker Dr. W. John Lough from the University of Sunderland, United Kingdom. Today it's my pleasure to invite such a great personality onto the platform. Now, before going to the session, let me take few seconds to give an overview about KL University. It is one of the prestigious deemed universities in India with NAC A++ accreditation and University Category 1 status and diamond ranking in QS International University Ranking sector. And coming to the KL College of Pharmacy, it was established 4 years back and a batch has been relieved in B Pharmacy. The courses offered include B Pharm, PharmD, M-Pharm Pharmaceutics and PhD programs. The highlights of the KL College of Pharmacy include we used to practice outcome-based education, Bloom's taxonomy, learning management systems, ERP, and we give special grooming for the students who joined here through their interest. Like they can select placements, for which we provide campus recruitment training and entrepreneurship skill training and higher studies training like GRE, TOEFL, GPAT, like that. And we also used to encourage them for Center for Extension Activities, sports, cultural programs like that. So here I can say there will be an overall development of the students who prefer to study at KL College of Pharmacy under the aegis of KL Deemed University. Here, let me give a small video of 1 minute regarding the KL University. Thank you for watching the video. Now I request our principal Dr. G. Chakravarti sir to give the welcome address for the today's international webinar.
So thank you very much, Dr. Koteshwar Rao. Good morning, everyone. Let me introduce myself. My name is Dr. Chakravarti, Professor and Principal, working in the College of Pharmacy, KL University. We have started pharmacy in College of Pharmacy in the year 2016, and first batch of the students came out this year, and they are reasonably placed in industries. From this academic year onwards, in addition to the B Pharmacy courses, we are offering PharmD as well as M Pharmacy Pharmaceutics courses. First of all, thank you for giving me an opportunity to speak to you, to speak to all of you. We are happy to conduct this session entitled Advances in Pharmaceutical Analytical Techniques in the field of pharmaceutical sciences. The main objective of this session is to know more advances in separation science and supporting methodologies involved in the pharmaceutical analysis field. I really appreciate Dr. John Lough, whom I associated 6 years long back in United Kingdom. to agree to give a talk and to share his excellence in the above mentioned areas. Thank you very much for all my faculty members in coordinating with the Mybo group and organizing the short event in a very short period of time. I wish and hope the interactive session will be helpful to the pharmacy students as well as the faculty members involved in the similar domains. Apart from this above session, Dr. LOUGH will also speak about DDD, which is a drug development and discovery program, which is a one-year postgraduate program in the University of Sunderland, United Kingdom, which was established long back. So once again, arranging a webinar on a core analytical techniques and advances in techniques and arranging and organizing in a short period of time. I thank you very much and thank you one and all for participating in this event. Thank you.
Thank you very much, sir, for your brief welcome note. Now I take the opportunity to invite my colleague, Dr. A. Rajashekar Reddy, to give introduction for our today's speaker, Dr. John.
Good afternoon, dear participants, and good morning, Dr. John. Myself, Dr. Rajashekar Reddy, working as an assistant professor in the College of Pharmacy, KL Deemed to be University, Guntur. It is my privilege to introduce today's esteemed speaker, Dr. W. John LOUGH from University of Sunderland, United Kingdom. Dr. John is currently working as a reader in pharmaceutical analysis and acting as a program leader for MSc Drug Discovery and Development, University of Sunderland. Dr. John is very closely associated with the University of Sunderland's highly regarded BSc Chemical and Pharmaceutical Sciences course. During the period of modernization of teaching, Dr. John has played a major role in the continuation of pharmaceutical sciences at undergraduate level through BSc Biopharmaceutical Sciences and designed and now leading the MSc Drug Discovery and Development course. And coming to the education of Dr. John, Sir has obtained his graduation from University of Glasgow. Later, he pursued his doctorate from University of Strathclyde, Scotland, United Kingdom. And coming to his experiences, Dr. John has worked as a postdoctoral fellow in the Department of Chemistry, University of Edinburgh, on ion-pair HPLC chromatography. Later, Sir has moved to the city of University and worked as a postdoctoral fellow there. To his credit, Sir has 8 years of industrial experience as senior scientist in Beckham Pharmaceuticals. During the time, he acted as a leader of chromatography team supporting drug discovery and preclinical drug development, working on drugs such as nabumetone, chromacalin, and paroxetine. And later, Dr. John moved to University of Sunderland with the designation of reader in pharmaceutical sciences. Even in his busy schedule of teaching and associated administrative duties, he published more than 80 research publications. He given more than 100 invited external presentations, and he completed around 31 successful PhD supervisions and editing or co-editing 5 books published by Springer, Pergamon, Wiley, and other reputed publishers. Dr. John have obtained around £750,000 in external research funding and made contribution to successful departmental funding bids in the excess of more than £2 million. Sir is a long-standing member of the Chromatography Society and acted as a president for the same for 2 years, and he is an expert member for the British Pharmacopoeia Expert Advisory Group. He's delivered short courses nationally and internationally in various countries like Canada, United States of America, Brazil, and India too. And he consulted widely in the UK and acted as a chairperson for chiral patent cases in North America. He has served as editorial advisory board member for 4 international journals and currently referee for 10 journals. Sir research interests are currently chiral liquid chromatography and selectivity in liquid chromatography for drug impurities and phytochemicals. With this brief profile, I would like to welcome Dr. John LOUGH to take over the session.
Okay, thank you very much for your most generous welcome. You almost know more about me than I know. Yeah, I'll just add that amongst you said that I'd kind of visited India before. I'd just like to add that my first visit in India was 1991, way back then. And over the subsequent 20 years, I actually visited India 17 times. So I've got great memories from that time, and hopefully I'll be able to visit again soon. So I'll... Good morning, of course, from me. It's actually afternoon with you. But I'll just start by showing you where Sunderland is in the UK, because you may not have heard of Sunderland. I kind of work at the university at Sunderland, and we've got good facilities too. I was very impressed with your facilities. One thing you won't find at Sunderland is a lovely cricket pitch and a chocolate room. So when I go back into work on Monday, I'll be demanding that we build a chocolate room. So this is where we are, bang in the centre of the UK. We're way up in the northeast of England, but so UK-wise, we're right in the middle, and that's where I'm broadcasting from, upstairs in my lovely little 4-bedroom detached house in Sunderland. And you are about 5-6 hours flight over to the right here. Okay, so that's where we're coming from. So the title, as we know, is Advanced Pharmaceutical Analytical Techniques. And it struck me when I was contacted just a couple of days ago to give this lecture. Well, what do we mean by advanced? Because for a second-year pharmacy student, advanced might be UV spectrophotometry, but for researchers in India, advanced will be way beyond that. So I'm going to try and cover as much as possible so that hopefully there's something for everything who happens to be out in the audience there. So we'll discuss advanced pharmaceutical analytical techniques. There are kind of a couple of contexts, but when we teach pharmaceutical analysis here in the UK, we talk very much about how it's used and vital for the pharmaceutical industry. So we're looking at how analysis supports this process. This is an old slide. I think it's probably taken from AstraZeneca. Maybe the time scale's a little bit more quickly now, but you have to do a lot of basic research before you start generating compounds. Then you have to make an awful lot of compounds to get one to the market. This is the FDA, the Food and Drug Administration in the USA, which is still probably the biggest market for drugs. And all through this process, from drug discovery, where you're kind of making lots of compounds and testing them, then you go into the preclinical phase. We call this the development phase, preclinical development, and then you're doing toxicology studies, doing formulation development, chemical development, and most importantly, getting safety data so that you get an IND, which allows you to start clinical trials and going through the different phases and getting enough information here to convince the authorities that your drug has got good quality, safety, and efficacy, and is suitable for going on to the market. The role of analysis is perhaps more set out more clearly here. We always talk about analysis being at the hub of all the activities in pharmaceutical R&D, and so this is like a wheel going from when you first start your research here, pre-discovery. I remember being in a project when I was in industry, going from the beginning of drug discovery to preclinical development. Might take 2 or 3 years, but there's a project set up for chronic organic brain disease, which covers Alzheimer's and all these kinds of neurodegenerative diseases. And they were given 10 years to find out, because a lot of the biology wasn't known. So there's support for analysis there. So now, if you're working analysis, you're interacting with all these different parts of the cycle. And so it helps to know analysis, even if you aren't analysis. And in today's industry, organic chemists and such like might find themselves doing their own analysis. So it pays to know about it. But the key thing about the role of analysis is that when you've got active compounds, you take out a patent here, and then you go through preclinical development, clinical trials, you get on the market. So from here to here, your patent lifetime is 20 years, and it might take about 12 years to here. So if you're lucky, you've got 8 years to have exclusive rights to your invention and sell your drug to the market at a decent price so you could recoup your research cost. So all this, you've got interactions with analysis. So if analysis is slowing things down, it will take you much longer to get on the market and you'll have less time to make money. And also analysis contributes to these, and you find things out in analysis which contributes to that, and often you come out with a better drug after you've done some analysis. So it's absolutely vital in pharmaceutical R&D. It is also vital as well to pharmacists once the drug gets in the market in terms of quality control. Okay, so it's all about helping industry get drugs to the market more quickly. Now, the other context is in when we're thinking about what is advanced is when we're talking about students, what is advanced for students. Well, this is the course that I'm program leader for. We have undergraduate programs in pharmaceutical sciences at Sunderland. We teach analysis in that, but this is the program I'm the leader for. And we started in 2009, 2010, and for the first 2 years, about 2/3 of the students came from India. So I think after that there were changes in the student visa regulations, but that's been relaxed now. So it'd be great after this kind of broadcast if we had lots more of you from India deciding to come back and join up this course. It's about drug discovery and development, but there's, as we'll see, and it's industry orientated. And if you go to that website, you could find more about it. And you'll see a horrible video of me. Okay, so on the course, this is the analysis we do in the course. And we talk about, so I'm going to talk about what's advanced later on again. But on the course, we assume that, say if you're a pharmacy student from overseas, as many as the applicants are, you might be familiar with these two, but we assume that people don't have a lot of knowledge of these basic techniques. So there's 3 stages to the MSc, and we cover all these at stage 1 in the first 3 months of the course. Okay, so qualitative analysis and quantitative analysis. So these are all the fundamental techniques that we use, and we cover all of these. And when we get to the next stage, we talk about, there's a module about science and technology for pharmaceutical development. And in that module, we've got some more advanced techniques, and there's chiral LC. A lot of it is separation science driven. I'm a separation scientist. I do chromatography, and NMR, mass spec are really, really important, but there tend to be 1 or 2 experts running these techniques. But when it comes to employment and jobs in the industry for analysts, a lot of it is to do with liquid chromatography. So we cover a lot of that on the course. We also do capillary electrophoresis, which is another separations technique, and thermal methods of analysis, things like differential scanning calorimetry, thermogravimetric analysis, and we look at sort of sample cleanups techniques, and we also look at hyphenated techniques. We put mass spectrometry on the end of gas chromatography and liquid chromatography as the detector. Okay, so in this, we always teach it the context of the kind of samples you find in pharmaceutical industry. So We talk about the API, that's the drug sample itself, the active pharmaceutical ingredient. We apply all these techniques to all these tests. If you look at a monograph or in a drug specification in industry, these are all the tests. Well, these are the main tests you've got to do. So we talk about how these are applied and how you choose the right technique and how you validate a method to make sure it's suitable for its intended purpose. Also, we'll look at the drug product. That links in with our formulation studies. There's a lot of formulation on the course. And there are more tests here. And again, you've got the drug and excipient, so these tend to be a bit more complicated in their sample preparation, but and also the choice of techniques can be slightly different. So we go through the techniques, strengths and weaknesses, and therefore which one is the best for each of these tests. And then more complex still is the drug and the biological fluid, where you've got low concentration of drug, you've got a very complex matrix, you've got drug metabolites, you might not have very much sample. So that's very challenging in terms of separation and detectability. So that's where some of these hyphenated techniques like LCMS come in, and they need to do more on sample pretreatment. And also more recently, a lot of the drugs going onto the market aren't small molecules, but they're large biological molecules like antibodies, vaccines, therapeutic proteins, maybe peptides. So the choices of technique and how you carry out the technique have to be different. So we cover all of this in the course. It's not just the techniques, it's about how you apply them, what's the best way to apply them. So you can't be thinking too much about advanced techniques without thinking about the actual context and what problems are posed by the context. Okay, so I've got some circles here. That's to do with the course, and that's where we where we cover them on these. We kind of teach these, but this is how we assess them on the course as well. And on the course, you get hands-on experience with all of these techniques, which is something you might not get in any other university. Who are really well equipped. Now, this is setting out the advances in analysis I'm going to address today. Coming from an undergraduate course overseas, you'll be familiar with titrations, and you're very precise, as is UV, but they are lacking in something called specificity. They don't actually, you're measuring everything in a sample that absorbs UV, and that won't just be the drug, there'll be other things as well. Similarly, if you're doing an acid-base titration, you're not measuring the drug if it's an acid, you're also measuring all the acidic impurities. So we need all these other techniques, and drugs tend to be in volatile compounds. So my main specialist area is HPLC, and because we could separate all the compounds out from one another on the column, then do the measurement, that makes HPLC more specific. So it's very, very valuable in the analysis of drugs. So going on to more advanced techniques, again, these are some of the ones we teach on the course. So you could have chiral LC to separate enantiomers. And when I talk about modern LC, I mean highly efficient types of liquid chromatography, which give you very short analysis times, because remember, time is money in industry, so you need to get all these samples analyzed very quickly. Capillary electrophoresis, again, another separation technique. It was very big in the early 2000s, late 1990s for small molecules, but its interest faded in that, but it's finding a place now in analysis of biologicals. Thermal methods of analysis, very useful in pre-formulation studies. NIR, near infrared, is kind of, for quantitation, is better than infrared, and that finds a lot of use on production quality control. Your solid phase extraction, liquid extraction are absolutely necessary when you're dealing with biological samples, to give you some cleanup before you do your chromatography, and also to give you a concentration step. Now, NMR is used for getting the structure of drugs, but in industry now, it's used increasingly quantitatively in drug discovery as an alternative possibly to HPLC. Not in your QC environment, because NMR is quite costly, but in drug discovery, quantitative NMR is increasingly common. And two-dimensional NMR, like with carbon-13 and proton, it's another two-dimensional NMR techniques are very good for giving you even more information about the structure. And as I mentioned, these are really needed for drugs and biological fluid, but now they're being applied more widely to API and drug product samples as well, because the specificity for mass spec detection kind of is very helpful. So going beyond that, these are things, we've got some more advanced techniques, which even an analyst would call advanced. And because of the range of people likely to be listening today, I'm only going to cover one of these. So we're talking about advanced. So I'm going to briefly introduce HPLC and talk about these two and how at a more advanced level, something we're just working on just now, these come together with modern LC includes UHPLC. I'll be explaining what that is, and chiral UHPLC, and HPLC and superficially porous particles. So that's what we'll be covering, but lots more. In terms of to cover all of the advanced pharmaceutical analysis techniques would take me a whole MSc course, but I've listed some of these other things very, very quickly. You use process analytical technology for monitoring the process and manufacturing. And these could be some quite simple techniques like acoustics, but it might be as simple as measuring pH or measuring temperature to make sure it's within specification at a point in a reaction scheme. And you do two-dimensional liquid chromatography, but in Europe now, people are getting millions of pounds for research into 5D-LC, which is really good for very, very complex mixtures, like protein digests, for example, and doing these really quickly. When you're developing HPLC methods, now companies like Pfizer believe you could do all this in silico on a computer without doing any LC experiments. They claim to have done that, but it'll be a very simple example, and I doubt whether that's going to, people are doing a lot of in silico method development to aid their method development, but I can't see it being totally in silico for some time to come. And in the spectrometry, the Raman imaging of proteins, and you get solid state biological NMR and single cell mass spectrometry. So these are big money techniques. You need to be a GSK or an AstraZeneca or a Novartis to be able to afford these. And a lot of your generic pharmaceutical companies wouldn't be using these techniques. So that's the whole range from simple ones to the normal ones that are used to kind of more advanced ones and the kind of really advanced ones. So we're going to talk about briefly this to lead into this, and we'll talk about chiral UHPLC, which is still a pretty recent development. Okay, so HPLC, apologies for those of you who know a bit of chromatography already. This is a simple modular instrument from the 1990s, so you can see the different parts. Essentially, we've got a column here, and on the column you put a mixture. The constituents of the mixture adsorb in the column to different degrees, and they get separated, and when they come up, they're measured by a UV detector. So for this, we need a mobile phase reservoir. So it's a liquid that's going to go through the pump. We need a high pressure pump. The column contains small particles of adsorbent, so it's a very tightly packed bed. So you need high pressure to drive it through. It's not like the big glass columns you still see in some organic labs where you use a gravity feed and just pour the eluent, your solvent down from the top, and it goes with gravity through to the bottom. You need a pump here, and that has to deliver a pulse-free flow continuously. So it's quite difficult to get good at pump design. You need to be able to inject under high pressure. The top of the column is at high pressure, and the bottom is at atmospheric pressure. quite a pressure drop across the column, but when you inject, so you need something that's going to inject something on the sample. If you're doing analytical work, that might be about 20 microliters or so, might be what you might inject. So you've got to get that reproducibly with very high precision and accuracy onto the top of the column. So again, it's quite a design challenge. The column is stainless steel to withstand the pressure. Typically use UV detection because just about, there are drugs which don't absorb UV, but not too many. And there's the waste reservoir, and there's a PC, and you can get big computers driving hundreds of HPLC systems, but nowadays most of them have got a network of PCs all linked maybe to laboratory information management system. So as you'll see in a photograph later on, things have gone way beyond this. That's the basics to help you understand what the basic process is, but now you just see these big towers of grey-white boxes, and it's difficult to know what's going on unless you kind of open all the doors and have a good look into it, which we do in the course. We've got videos of the course we use at the university where you do a little tour through these modern HPLC systems. That's a basic system. That's how it works. Okay, so to understand why we need chiral LC and UHPLC, I'll talk about the fundamentals of the separation. Essentially, at the end, when the two compounds are separated, this what you see is a chromatogram. And you want, say you want to measure this one, you have to able to separate this peak from other compounds in the mixture. Maybe one's coming off afterwards or just before. You can see this is the more closely eluting one. And you want baseline resolution here so that when you measure this peak with the UV detector, you're measuring this peak and only this peak. So the key thing in chromatography is to get resolution. And you can get resolution by increasing the retention time. That increases your analysis time. So you want big, reliable baseline resolution. So this looks like, okay, that's almost baseline resolution. But if you're making up the mobile phase every day and using a different batch of the column you're using, you may lose that. So you want reliable baseline separation. This is a resolution of 1.0, so typically you'd want 1.5. So it's all about getting resolution quickly. And as we'll see, I think on the next slide, you could actually increase retention, you could increase selectivity, the differences in their affinity for the column, and also talk about increasing efficiency, which is the key driver for improving going into modern chromatography. So these are poor separation. This is what you want. In fact, you want better than that. You want clear baseline resolution here, but you see if we've got poor retention, we want to get the separation. If we've got poor selectivity, we want to get the separation. If the peaks are very broad, it's actually very difficult to separate them. So what we want is to get these peaks narrower so that they're easier to separate. So the narrower the peaks are, the more peaks you could separate per unit time. So if we make these kind of infinitely narrow, you could probably get hundreds of peaks long here. So that's what we're aiming to get to increase throughput in chromatography. So it's about getting this peak narrower. Okay, so as there are processes going in the column, you get separation, and there are processes taking place inside the column, which give you band broadening. You just get, say, if you put 20 microliters at the top and it goes down the column, it's going to spread out. It goes in and out of the particles, and it does that at a different time. That's called mass transfer. And also you got a bed of particles, so they take different pathways. So all these three processes called band broadening. So when you come down the column, you don't have a infinitely narrow straight line here. It's spread out. And because some go faster, some go slower, some come off more quickly than the mean, and some come off more slowly. So you get this Gaussian distribution. This is what a chromatogram looks like. So, and as a Gaussian distribution, it's characterized by the standard deviation. So when you're trying to reduce the band broadening, you can't, if you thinking about reducing band broadening, you can't just take the standard deviation and add it up, because some might be positive, some might be negative. So what you do is, as in statistics, is sigma squared. And sigma squared is the variance of the kind of error in the pathways coming down the column. And L is the kind of length of the column. And so that's directly proportional. The longer the column, the more the band broadening will be on the column. So it's all about reducing that. So to reduce that, the variance is additive. So H, we call this the height equivalent of a theoretical plate, needs to be reduced as well. So we look at the individual causes of the band broadening and just add them up. So if we reduce these three processes of talking about, eddy diffusion, longitudinal diffusion in the mobile phase, and mass transfer, if we introduce these, we could get very, very sharp peaks, okay? And therefore separate a lot more compounds in the same space of time. So to do that, you need small particles. You have to use, we'll talk about the Van Deemter plot in a second. You have to get the right flow rate to minimize the longitudinal diffusion in the mobile phase and to minimize the mass transfer. So we'll have a look at one of these plots. You have to get the optimum flow rate, but also you want a uniform bed, and you want no kind of gaps in here, and you want the mass transfer in and out of the particle to be very quick. So you want small particles in a uniform bed. And the best way to do that, get them very densely packed, is to use small, spherical, totally porous particles, because the chromatography takes place inside the particles with a narrow particle size distribution. If you have a kind of big one and a small one, you can't pack them in a nice even bed. So 5 micron particles were introduced in 1978. And for about 30 years or more, that's still the kind of standard particle size that people are using. So why is this? Why are people still using these 5 micron particles? So 30 years plus on, and that's because you go to very small particles, you get the columns become more difficult to pack. It's difficult to get porous frits to retain them in the column, and the increases in separation you get by going to higher and higher in efficiency. The resolution is proportional to the square root of the efficiency. So up until the early 2000s, the effort needed to get much better efficiency simply was too difficult to achieve. But that did change in 2004 with the introduction of sub-2 micron particles by a company called Waters, and that was UPLC. So they put a trademark on UPLC, so all the other companies who couldn't do that, and instead of operating at a maximum pressure of 400 bar, the equipment introduced kind of operated at 1,000 bar, so at much higher pressure capacity, and also to be able to handle much smaller volumes, much narrower bands. You need much faster electronics, and the volumes outside of the column have to be very small. So by introducing this UPLC, kind of Waters got way ahead of their competitors and were able to do much faster, more efficient chromatography with their new instrument, the Waters Acuity, which was launched in early 2004. Now, the other companies couldn't achieve these high pressures and that improvement in instrumentation and column hardware. So what they did to compete, as we'll see in a later slide, it took them about 5-6 years to catch up. But they were criticizing UPLC because the need for ultra high pressure all the time. So what they introduced, so it could be used in an HPLC system, is what we call superficially porous particles, where the particles are larger, but you still, and because this is non-porous, you still get the kind of the residence time in the particles is still very small because you've got this thin layer. Instead of using a much smaller particle, you've got a very thin layer here. So the residence time in and out the stationary phase is much quicker, and therefore the mass transfer is much lower. So this gives you almost like UHPLC performance, but you could do it on an HPLC system. So it was UHPLC versus SPP for about 6-7 years until everyone could do UHPLC. No, UHPLC, that's what the other companies called it because of the UPLC trademark. And then everyone started using UHPLC, but these still have their place. And the very latest development is by Waters, who've actually introduced a sub-2 micron particle. Phenomenex also have one, which is sub-2 micron particle, which is superficially porous. These have their disadvantages, as has UHPLC, but they've kind of transformed the speed with which we could do chromatography. And again, remember, get things to the market. So this covers some of the things about the reasons why both these work. And this is them on a kind of different scale. Okay, so the low intraparticle diffusion path length, that improves the mass transfer, the C term in this Van Deemter equation I was talking about. So you get much faster peaks. Okay, this is the Van Deemter plot. It's the HETP I was talking about, that proportionality constant between the variance and the column length. You want that to be as small as possible to get as many theoretical plates in the column. So we're looking at it to be low as possible. So this is 5 micron particles. You operate down here to get a low H, and therefore we get a high column efficiency, lots of plates in the column. And when you go to 3 micron particles, it's better. But when you go to 1.8 micron, not only, now this is a big improvement in efficiency, and it really helps the resolution. But the key thing is, because we've improved the mass transfer in and out of the particle, this C term, which is the mass transfer term, is much flatter. So it means you could go to really, really high flow rates and still get high efficiency and still get really sharp peaks. Whereas in conventional HPLC, that doesn't happen. The peaks get wider as you go to higher flow rates. So you go higher flow rates, you get much higher back pressure, but the UHPLC systems actually can handle that. Okay, so nowadays, this is our separation science laboratory, and these are individual HPLC systems. So these are our MSc students, very multicultural. We've got a lady from Bangladesh, a lady from Greece, and a lady from Nigeria. And that's the back of the HPLC systems. This is a kind of a mass spec, believe it or not, as a detector. So we've got 8 HPLC systems and 8 GC systems, and each one of these is UHPLC, and some of them have got different types of detector on. So all our undergraduates, never mind our MSc students, get hands-on experience on these. And if you've been operating these as a student and you are looking for a job, and you could say you got hands-on experience of using an Agilent 1290 and using the ChemStation software and done method validation, you're really well placed to get a job in the pharmaceutical industry. Okay, so we started using this ourselves, not till 2008, because when UHPLC first came out, only the big companies could afford it. And I'd worked in industry, and one of the compounds I'd worked on was paroxetine. And so I've got fond memories of paroxetine. Not that I suffered from depression, but that's paroxetine is sort of an SSRI, a selective serotonin selective reuptake inhibitor. So it's in the late 80s, it was a kind of new type of antidepressant drug, which is a major advance on the ones that had been used up till that time. And I developed I was the first person to develop HPLC methods for this compound, and I knew it was quite a challenge. The paroxetine is the minus, this is trans stereochemistry here, and you get the enantiomer, which is trans stereochemistry with this into the plane, this out of the plane. So because the serotonin receptor is very stereoselective, this went onto the market as the single enantiomer. But never mind that, at this point, we had to separate it from the cis impurity and the when this is a proton, all these fluorinated drugs, you always get a desfluoro impurity, about sort of 0.3, 0.5%. So we had to do that separation. So that, I know this compound well, let's just take the method, an HPLC conditions, and we'll ramp up the pressure and we'll do it much more quickly. Well, we couldn't actually do that because the method we'd used in industry had been slightly different. And just taking reverse phase HPLC and ramping up the pressure and flow rate, we could separate from the desfluoro, which I recollect being the most difficult of the impurities to get the separation from. But that was using something called ion pair chromatography, which we won't go into. It's another mode of LC, which is slightly different from reverse phase HPLC. You've got a C18 column and a aqueous, polar aqueous mobile phase. But when you're not using ion pair, the most difficult separation is the cis one. So when we just do regular HPLC conditions and ramped them up, we didn't separate from the cis enantiomer. So you have to still think about the method development. It's not like taking conditions, getting high efficiency, bang, you've got the separation. You still need to think about the selectivity. So I recollected then that our colleagues in the manufacturing site had done something different because they didn't want to use ion pair chromatography. And they used, to get the separation from the cis, they used about 3 or 4% of tetrahydrofuran in the, so yeah, we needed 5% THF. So we said, okay, let's do the, and that manufacturing method had gone British Pharmacopoeia. So we looked at the British Pharmacopoeia mobile phase and gradient, and that gives you a 60-minute run time. So we did this in stages. We upped the pressure. Then these new materials for UHPLC are also very temperature stable as well. So if you go up to higher temperature, the mobile phase becomes less viscous. So for the same pressure, you go at a much higher flow rate. So we went up to the details are 80 degrees. And again, the flow rate will be, yeah, the flow rate is 1.5. But remember, there's a very short column and a 2 millimeter ID column. So for that, this is a phenomenally high flow rate. The volumetric flow rate, remember, they're very narrow. The internal volume of the columns is much smaller. So by doing that, just speeding everything up and ramping up the pressure and temperature, this is the separation we got here. This is the cis, sorry, it's not. This is the cis in here, tucked in between the desfluoro and the paroxetine. So instead of having 60 minutes, we're able to do that in 2 minutes. And we talk about 2 minutes, but all the separation's done by here. That's one of the other impurities, and there's another impurity here and here. So it wasn't just these difficult ones. We're able to separate all the impurities in about 1.2 minutes, and the rest of the time is just re-equilibration when you're doing a solvent gradient and changing the percent organic to get the less polar compounds to come off the column. So that was our first foray into UHPLC, and we've been using that ever since. Now, another area, which is an advanced area of separation science, has been going for a while, is to separate enantiomers. Now, the enantiomers of drugs are mirror images. And then solution in water or melting point, infrared, NMR, they're identical. So if they're identical, you say, well, why bother separating them? But in a biological system, they're different. And this is why they're different, because biological systems contain proteins, which contain L amino acids and D sugars. And here's what happens if you approach these two mirror image formed with another chiral molecule, like an L amino acid. This is the mirror image. We just spin it around to make it more obvious what's happening here. If you have 1 point interaction, like Y to D, it's just the same. You can't tell the difference. There's 2 points of interaction, adding ETX, you can't tell the difference. But if you try and make a third point of interaction, W to A, you can't do that here in this second case. So these could be negative interactions, but if these are all positive interactions, this one will interact more strongly. So this single chiral molecule, like say L-amino acid, could distinguish between the two enantiomeric forms. Now that happens in the body, so you get different pharmacological activity. All these processes in the body are mediated by proteins, which contain L-amino acids, D sugars. So your drugs go through all these different processes, and each of these processes, you've got potential for discrimination. So when the drug acts in the body, going through all these processes, it'd be a miracle if the two drugs acted the same. In fact, they often act very different. The classic case, which brought about all the emphasis on safety in the pharmaceutical industry, was thalidomide, where babies were born with truncated limbs, and this was a sedative, and it was the R was the active form, and the S was the enantiomer, which is giving rise to this phocomelia. And that's quite a major case, and that so the regulatory process of drugs was really tightened up after that, and it brought about a really big interest. That was the early 1960s, and it brought up a big interest in the field of drug chirality. Now, it's not always as extreme as that. There are some cases like ethambutol where one enantiomer is not active but highly toxic, but often it's a case of they've just got the same effect, but one much stronger than the other. They could have different activities, or one could be totally ineffective and harmless. But if you've got a new drug and half of it's harmless, there's no need to deposit it, put in the body. So for all of these cases, the regulations kind of changed in the 1990s. So essentially now, if you're looking at a small molecule and it's chiral, you've got to investigate both enantiomers, and almost always that ends up with putting one of them on the market rather than a 50-50 racemic mixture of the two. Because up until the mid-1980s, most of the synthetic drugs on the market that were chiral had actually been racemic mixtures. Okay, so you need to be able to separate them. You treat them like other impurities. So if you've got a chiral drug, the other enantiomer is an impurity. So you need to be able to separate them. And without spending my usual 1 hour on the history of chiral liquid chromatography, these are the early chiral selectors who use all this. This was quite a challenge, actually, because the thalidomide scare was early 1960s, and it wasn't until the late 1980s till all these chiral selectors immobilized on HPLC columns became kind of commercially available. And the obvious one was proteins, because proteins just do the distinguishing in the body, so why not put them in an HPLC column? Now that was challenging because they're big molecules, it's not good for the mass transfer, and they weren't chemically and physically stable, but they were very good for separating a wide range of enantiomers. So that was the late 80s, and that revolutionized things, and companies started producing single enantiomer drugs. But they needed something that was as good for chiral separation as the proteins, but was much was kind of more stable and not quite so big. So these are the various initiatives that took place in the 1990s, and as a result, the different colors are just different manufacturers and different types of chiral selector. But there were major developments in the early 90s, and we got chiral stationary phases. Primarily, these ones which worked in reverse phase, these are also known as synthetic multiple interaction chiral stationary phases, and derivatized polysaccharides. So these became the most popular. So these were more physically and chemically stable, and were smaller and complex enough to give you a wide range of chiral separations. So by the late 80s, you could have just 2-3 columns like these, There's a polysaccharide. This is the polysaccharide chain. And if you put R groups on instead of OHs, and you link with a carbamate here, you can actually get very good chiral separation. So instead of having to do a lot of chiral method development, which is what I did a lot of between the mid-80s and the mid-90s, you could just have 2 or 3 of these columns. And this is another one, the welco column, which is synthetic, not relying on the sugar molecules. We got 2 chiral centers here. And you got the electron-rich pi system, electron-deficient pi system, and that's almost like a cavity in here. So like the derivatized and mobilized polysaccharides, you could get really good stereo selectivity and separate a wide range of enantiomers. So much so, instead of doing method development, you just get 2 or 3 columns and you screen them, you take the best. This is a schematic from Pfizer, and you'd put your chiral, your mixture of enantiomers down these 4 different chiral columns, and you'd see which one was best with 2 or 3 different mobile phases. With these intelligent systems with the PCs, they would then take the best separation and optimize it overnight. So all this could be done automatically. So as the mid-1990s, it's like all your chiral HPLC problems have been solved. And kind of getting into, this is work done by our MSc students actually. We kind of did a 4-column screen. We had 3 different welco columns. We were trying to pick the best one because there's slight differences in chemistry there. So we just screened these 4. We used 4 mobile phases. We used 5 mobile phases, but we're going over the top. We could have probably got there with 2 mobile phases. And this was these legal high drugs, and came out in around about 2010. And these were all chiral, so we thought that we had to look to see whether these were racemic drugs or whether they're single enantiomer drugs, which enantiomer. So the earliest and most popular of these was a drug called mephedrone, which is similar to an amphetamine, and we tried to do chiral separation of that. So this is the welco again. Don't have the structure of mephedrone, but it's like amphetamine, except it's got a carbonyl instead of CH2. Not only would get a good separation with very high resolution, we're able to do this so well. We've got such a good separation that we're able to, normally on a HPLC column analytical scale, you'll have about a nanogram, a couple of nanograms on the column. But here, we were able to overload it, and the peaks get wider when you overload it. We're able to put 2 milligrams on the analytical column and get enough to do subsequent testing. So we stopped there because these drugs are illegal or had become illegal. So the reason we stopped there was, had we started collecting these peaks and isolating them, you needed a license to do the analysis, but we didn't have a license for making new illegal drugs. So if you make a single enantiomer drug here, it's like a new drug. So, and also by publishing this, we were a bit worried on the ethical side of things that we're telling all these people in these illicit laboratories in China, not India, mainly in China, how to actually make better illegal drugs. So we thought we better stop there. But it just shows you how easy chiral LC had become by that time. So what is there left to do? Well, we could do it quicker and faster, but there's more to it than that. So this was chiral LC. This is UHPLC. And all the other companies catch up a lot of time later. But there's a big gap between UHPLC and chiral UHPLC. And that's because the chiral selector interacting with the chiral drug with 3 points interaction is a fairly strong interaction, and the mass transfer is slow. So you can't get quite get improvements in efficiency. So that you'd get from a typical reverse phase HPLC column with a kind of alkyl bonded silica stationary phase. So these became available, but it wasn't really till about 2017 till a lot of them were coming out. There was just one there, and this didn't really, big companies weren't buying them in a big way until about 2017. So people who were in industry were doing it quicker and faster. We all thought, well, okay, so what? But if you got a good chiral separation, you could do it quicker and faster in a normal column. But what we wanted to do, if you look in the pharmacopoeias and in industry, you want to save time. So in perhaps should have a slide for this. If you're trying to become more efficient and do things much more quickly, you need to test for the drug impurities, which often takes, if you think of the Paroxetine example, takes about 60 minutes. And we could do that in 2 minutes, but for a drug, you get the assay, the purity, you get the related substances, the structurally related impurities, which is might take 60 minutes. So but you have to do another test with a chiral column to do the to get the enantiomeric impurity. So we thought, well, doing chiral LC quickly is a bit boring. Anyone can do that. So let's see if we could go beyond that. Let's see if we would take a chiral column and use it to do the structurally related impurities. We'd had done something of that nature back in 2012, but it's a fairly one-off example. But we thought with these UHPLC columns, maybe we could do that all the time. Despite these chiral columns having a reputation for not having good achiral selectivity, well, we thought, well, let's just take a look at that. And because you're looking at all the impurities, the example I showed you of methadone uses so-called normal phase solvents like hexane and dichloromethane and maybe a trace of isopropanol. But most drug assays and related substances tests are done in reverse phase. So we looked at these macrocyclic antibiotic chiral selectors, which are almost like miniature proteins. They're smaller than proteins, but they've got peptide chains in here, small peptide chains, and they've got sugar molecules. So these are quite effective in reverse phase and something called a polar ionic mode, where you've got a high content, you got kind of methanol and aqueous, but a higher percentage of methanol. So we thought, okay, well, maybe if we do this in reverse phase, we could maybe separate the drug enantiomer and the all the other impurities in one test rather than doing two separate tests, both of which take a long time. So we did that with a kind of chirobiotic called HPLC column, but the resolution wasn't quite good enough, so we decided to go to these not UHPLC columns. These are actually using the superficially porous particles I was talking about earlier. Fortunately, I knew the guy who invented these columns, so I went to buy one column for £2,000, which is a lot of money. This Vanco shell, which is the vancomycin chiral stationary phase we've been using, but with superficially porous particles to give you much higher efficiency. So I tried to buy these for 2,000. So for 2,000, they gave us 5 columns. So we thought we might as well look at these as well. But the main work was in Vanco shell. And so we've got, these are good chiral separations. And what we're doing, we've got the superficial porous particles, so we've got much better efficiency, shorter run times, but we're hoping that they could also separate the other impurities. We had some success with the chirobiotic V HPLC column. So we kind of, and also we're looking for racemic drugs where there's a genuine interest in the single enantiomer. And one of these, the best separation that is in the literature in bupivacaine, and this is at an analytical scale 0.1 mg per mil. And when you're working with related substances, you want to look at trace level impurities, so you have to put a much higher load. So we could go up to 1 mg per mil and still get really good baseline separation. Notice that this is an unretained peak, so these are coming off quite early. So, but we wanted to say, when you're looking at related substances and drug impurities, you have to kind of get them down to about level of 0.1, 0.05%. So we thought, okay, well, the chiral separations are easy. Let's see if we could pick up the impurities. So this is, we took the active enantiomer, which my memory escapes me whether it's the R or the S, but it had been an interest in that as a drug. It's called Chirocaine, which is made by a company called Chiroscience and licensed out to AstraZeneca. So this is a genuine thing, which is a genuine interest, not just of academic interest. And we could quite easily see when you load up 1 mg per mil, it's on the tail here, but that's very easy to integrate. We could pick up 0.1% very easy. So the quantitation we could do well. But with this example, this is a very good chiral resolution, but it's coming off too early for us to be able to separate 7 or 8 other impurities. So it so happened, I can't easily go back. I could, hold on. If we go back, we also had this one called Nicoshell, which was new. There wasn't an HPLC, so the nicotinic receptor. And that hadn't been as successful with Vancoshell. We thought, well, we've got the column. It looks almost as good. Kind of let's try it. And under the conditions we're using, and we tried drugs. We've got lots of drugs in our lab. And we tried drugs which other people hadn't kind of looked at on the Nicoshell and Vancoshell, because we've been studying chiral LC for years and years now. And there we tried 18 compounds. Actually, only about 3 or 4 worked on the Vancoshell, but about 10 or 11 worked on the Nicoshell. And one of them, well, we thought, there's some good separation. I thought we want one which is really genuine. There is an interest in the single enantiomer drug. I thought, wow, what about Paroxetine? So Paroxetine hadn't been one of the 17 or 18 compounds. We thought, well, let's try that, because that's something we've done before with UHPLC, see if we could do it with the chiral resolution. And okay, so you can't see this on my screen. So we tried it. This isn't a 50-50 mixture, because these are both sold as single enantiomers. You do have to do a test for the plus trans enantiomer. There's a nice separation, so let's see if we could do that and pick up 0.1% of that. So we had a mixture which not only had, remember, I was only asked to do this a couple of days ago, so I haven't labeled all these up, so I'll just take you through this. This is the Paroxetine at 1 mg per mil, and this is the kind of plus trans trace enantiomeric impurity at 0.1%. Okay, and this mixture also contained the desfluoro and the cis. We believe the desfluoro and the cis to be this one and this one, but at this level, we're picking up some other impurities. So, because there are some other impurities. So what we have to do now, and what I'm going to have our MSc students in the drug discovery and development course, incidentally, in September, they're going to go through this again and identify all these impurities. So things are looking promising that we could use UHPLC, superficially porous particles, to do drug-related substance testing very, very quickly. And if we use the chiral ones, it's looking promising that instead of having 2 long tests, we could have 1 short test for all of the structurally related impurities, including the related substances one. Okay, so going back to this, this is the separation science, and we're working in lots of different areas up here. But to get back to where we started, we've covered HPLC. These have been around for a while, but there's still a lot of people are fairly new to these. But if you combine the two together and using them for structurally related impurities, that's fairly new and fairly advanced, and with a bit of luck, we'll get around to publishing that before millions of other people do it. Okay, but if you're thinking about advanced pharmaceutical analysis as a whole, and you're going to work in industry for one of the big major pharma companies, these are some of the companies, other advanced analytical techniques you'd use, but you'd have to almost have a PhD and do a postdoc if you're going to be working these in industry. Okay, well, normally I go way over time, and I'll look to be 10 minutes over time, but let me first apologize and then say I wasn't really expecting such a wonderful and long introduction. So I'm happy if you've got time and you're not going elsewhere on a Saturday evening in India. I'm happy to take questions. If I could end the show. Yeah, good. I think we're out of that now. So I've been talking for an hour and I haven't been able to see anyone, so I hope there's still people out there. You still there?
Hi, Dr. John.
Hi. So
That's really
I'm happy to take questions now if you've got time.
Yeah. We have time for a few more minutes, and we will take the questions. No issue. And that's really an excellent session. You have given a very informative examples along with the case studies. So thank you very much for such a brief session. You tried to cover maximum advanced analytical techniques. Thank you very much. Now we will go
I should add, as well as advanced pharmaceutical analysis, do remember if you've got an MPhar, if you got a pharmacy degree from India, you still got time to join this year's MSc course in Sunderland.
Yeah, sure. Most welcome. I welcome all the participants also to have this session or to have the questions. Now we have with us one person to ask a question. I would like to add him.
Okay.
Please unmute your mic, Mr. Nagaraju, and please raise your question.
Okay, sir. Thank you. Good morning, sir. My question is related to the TLC, sir. What is the optimum RF value for isolated compounds, sir?
For thin, I thought you were going to ask, can you do chiral thin layer chromatography? The answer is yes. Any chiral selector you put on silica for HPLC. Now, the optimum RF value for TLC depends on what you're trying to do. If you got a complex mixture, you want the best possible separation of the compound you're interested in. So you'd have to use the complete range, pick a solvent such that, say you're using silica, you need to pick a solvent so the least polar compound goes right to the top with an RF of 1, and the most polar ones are sort of down at the bottom, and the one you're interested in will be in the middle somewhere. But if you're just monitoring a reaction, you might want it's near the bottom, so you could do things very quickly. But if you're going to use PrepLC, maybe I guess what's critical isn't the what's critical is probably not the eluting solvent, but the solvent you extract the drug from the plate with. So it all depends on what your application is. Okay, so there's no definitive, this is the best RF. But I mean, if you've got a TLC plate, you want to use as much of the plate as possible. But you don't want it running too close to the top, because I used to work in London, and we in the center of the city of London. These days, there's a lot more pollution with cars. We'd have the lab windows open, and over time, the TLC silica would absorb pollutants from the environment. So we'd have to run the solvent up the plate to clean up the plate before we used it. So I guess there's not so much pollution in India, especially during lockdown, but that's always good practice if you're going to do prep work and you're using something with high RF value that goes near the or that runs close to the top.
Yeah.
Thank you, sir.
Thank you, sir. Thank you, Nagaraju.
Okay.
Sir, we will pick up one more question from the participants. I will display on the screen. So
Okay.
it is from Manjiri Shinde.
Okay, fine. Are there any? Okay, well, I'll talk. Is there one is money. Talking in British terms, or can I talk in US dollars? Let's talk about pound sterling. And I think every time I go to India, the rupees to pound sterling exchange rate seems to have changed dramatically. But in pound sterling, reverse phase HPLC column, which is an alkyl silica, might cost about £200. A chiral HPLC column could cost about anywhere between £500 to £1,500, or £2,000 even for some of them. And your kind of superficially porous chiral columns cost £2,000. So I was very lucky to get, because I know the inventor, I was very, very lucky to get these cheap, and I was doing research on them. So that's the main thing. I think the other thing is the loadability. You lose efficiency when you put large amounts on the chiral columns. UHPLC, you need like a UHPLC cost them it costs £50,000, and we've got 8 of them, so that's a lot of money. And if you're using superficially porous particles, you can use a standard HPLC column, but you still have to be careful about extra column band broadening and to keep your efficiency. Okay, right. So, well, there probably is now the identification. Well, they can identify. You're getting into biologicals, and I do LC biologicals, but that's probably my weakest area. I think you'll probably find there is tests, but you wouldn't. I think you'd have some kind of affinity test or biological activity test rather than a chromatographic test. Now, you could do HPLC of antibodies. I'm not too sure about viruses. Okay. Is chiral LC important for all new compounds that are discovered? The answer, if they're small molecules, the answer is yes, because even if it's achiral, because if it's most new small drug molecules are pretty complex these days, so most of them are chiral, but even if it's achiral, when you get metabolism, you could generate a chiral center. So, and it's very rare for a new synthetic drug not to have any chiral centers in it. So I'd say yes, it's just about important for all new small molecule drugs. Yeah.
Yeah, we have one more participant, sir, for asking a question. Hello,
Okay.
Hello, Mr. Sami.
Hi, sir. How to join in the drug discovery course?
When is the drug?
How to join in the drug discovery? How to join in the drug discovery course?
On the drug discovery, but what are you asking about the drug discovery part? I mean,
course, sir.
Oh, the course, the drug discovery. All right, okay. Yeah, on the course. Okay, well, it's MSc Drug Discovery and Development. So on the drug discovery side, we talk about medicinal chemistry and things like combinatorial chemistry. So there's chemistry in that. And we talk about the biology, about proteomics and metabolomics, about...
Dr. John,
yeah.
Dr. John, sorry for the interruption. He is asking how to join the course. How to join
how to join the course. Oh, sorry. Well, you go on the university website and you just... My apologies. Sorry. You do it. Well, you could do it through an agent, which maybe helps, but you don't have to do that. You could go to the university website and you could apply online. There's an online application form, and what you have to... So if you've got a pharmacy degree from an Indian university or pharmacy college, that's the fairly standard entry qualification that most of them have. So if you've got that, it should be fairly straightforward. You go online, and usually what you have to provide is you need to get an IELTS score, and the score needed will be on the university website. It's not too demanding. It's much the same as other MSc courses, and also you need a personal statement and a couple of references and a kind of transcript or a degree certificate. So for this year, the rate limiting step will be making sure you got the IELTS and getting a visa in time.
Yeah, sir, we will take the last question, sir, for today's session. We will take the last question.
Okay, fine.
So, there is one more participant.
Okay, fine.
Your voice is not audible. Hello, Ramyasri, your voice is not audible.
She must be on mute.
No, we are unable to get. Will you please remove that mic, and will you please try it again? From the computer, please remove that earphones and try it. Speak out. No, sir. Okay, she has posted a question to us, sir, directly. So the question is like, what about the visa criteria? Which type of visa will be granted if we join in MSc Drug Discovery course in your university? That is her question.
Right. So Tier 4 visa, and they've changed it. We had a new Prime Minister in September 2019, and one of the first things he did was to change the visa conditions for overseas students. Now, there used to be a 2-year post-study visa until 2012, and then the government tightened up and that wasn't available, so it reduced our number of overseas students. But our applicants this year are much higher because the first thing, I don't know if you've heard of Boris Johnson of the Conservative Party, so he's involved in Brexit and all kinds of things, but the first thing he did was to relax the visa regulations and be more welcoming to overseas students. So you get a 1-year post-study, you're able to stay in the UK and get work experience or get a job for 1 year after study. So you could stay in the UK for 1 year after studying. And also, he's introduced a point system, and a lot depends on the kind of salaries you could get. So I would think that most people doing the MSc, if they get a job, they probably have enough points to apply to stay in the UK. Not that I want people to come and stay in the UK. I'd love you to come to our MSc course and then come back and feed in all your knowledge to developing India.
Sir, may I know the prospectus for the jobs after doing that MSc course in drug discovery? Is it for UK job application, or are we able to?
No, it's very good. We've got lots of. One of the things we cover in the course is the regulatory side of the pharmaceutical industry. So we've got a lot of our students who went back to India have become regulators, and they're doing pharmacovigilance. A lot of them have found drugs that way, and so almost the ones that come back to India, I think they're almost all gone in the pharmaceutical industry.
Yeah.
I do have data on that, but I haven't broken it down from country to country. I was still in touch with 2 or 3 of them and kind of LinkedIn. So the employment rate is very, very high. The only people who don't get jobs after doing our course are people who actually don't want a job, which seems strange. You do get 1 or 2 individuals like that.
So Ramyasri has also posted one more question, sir. She is asking, is the prior experience of handling HPLC is mandatory to join the course, or are we able to learn?
No, not at all. We appreciate that we're taking applicants from all over the world. So we assume we start right from the beginning and go up like that very steeply. So it's stage 1, stage 2, and stage 3 of the MSc course. And at stage 1, we make sure that everyone's at the same level. So you get your HPLC lectures and theory in stage 1. So it's we kind of assume that people don't have any knowledge. We assume that people might know a little bit about UV and titrations, but even then we still cover that a little bit, you know. So you don't need a lot of experience, but you'll get a lot of HPLC experience on the course.
Yeah, here is the last question, sir, from her. So after doing this MSc drug discovery course, is it an eligible criteria to join PhD also? Is it like a 2 years PG course equivalent, or it's just an additional skill-oriented course only?
Well, that's an interesting one, actually, because you can actually do a PhD in the UK, I think, direct from, say, a pharmacy degree in India, but just about everyone. But that's if you're self-funding. But most people are more comfortable doing a PhD after they've done the MSc. And if you've done the MSc, a lot of our MSc students like it so much they stayed at Sunderland to do a PhD. So I might have I won't get the presentation up again, but I do have a slide there about the percentage of what our students do, and I think 20% go on to do a PhD.
Okay.
And it's
so thank you very much, Ramyasri, for good questions. I hope all are answered. Thank you very much, sir. Dr. John, that's really an impressive presentation, and the answers to all the queries are also very informative. And the course, whatever you are running, the MSc in Drug Discovery and Development, it's really appreciable course, and I hope the students will make use of it, because LCMS or HPLC, the lab, how much sophisticated it is. So it's very impressive. So I thank you very much for spending your valuable time and sharing your knowledge to all the participants over here. We got the participants across the world from different countries, and it's really a wonderful informative session. So total 1,000 plus participants has registered for this event, and I am very much happy to share.
How
So
many?
on behalf of 1,000 members plus has registered for this course.
Okay, well,
for this webinar. Yeah.
Hopefully
So
we get
thank you
some
very
of them
much
joining
for
our
your valuable
MSc
time.
course.
Yeah. So
Okay, so
I will take more details regarding this MSc course in drug discovery, and those who are having queries, I will share to them also, and I will connect them to you. So please guide them for possible higher studies and skill development areas. So finally, as a formal vote of thanks on behalf of management, principal, staff, and students of KL College of Pharmacy, Koner Lakshmi Education Foundation, Vadeshwaram, Guntur, from India, I would like to thank all the participants over here for spending your valuable time. We hope that you have learned some of the informative things shared by our speaker, eminent speaker, Dr. John Lough from the University of Sunderland, United Kingdom. So thank you for giving me this opportunity. So thank you, Dr. John, for spending your valuable time.
I'm going off to do shopping for my wife now.
Thank you. Have a nice day. Thank you very much.
Cheers. Thank you very much. I'll leave the studio now. Okay, thank you.
Yeah, thank you. Thank you, one and all. Meet you in the next webinar by KL College of Pharmacy. Thank you. This is Dr. GSN Koteshwara, Associate Professor at KL College of Pharmacy. Thank you.